Hi everyone, my name is Fred Rodriguez and I'm a member of the Tretyakova lab from the Medicinal Chemistry Department. Today we'll be talking about the primer extension assays. So these gel assays are actually quite important in our lab as we can study the effects of perturbations of DNA structure related to DNA transactions such as DNA replication or DNA transcription. So for today's experiment, let's kind of talk about what's going on. We start off with a radiolabeled primer. This radiolabeled primer is a Nucleotide template DNA in order to make a DNA duplex that can be recognized by DNA polymerases for either transcription or replication. Whenever you run them on a page denaturing gel, what happens is that the DNA primer and the DNA template come off, and the only thing you should be able to visualize on these gels is a radioactive DNA. This radioactive DNA, when it's mixed with its polymerase and deoxynucleotide triphosphates or nucleotide triphosphates if you're doing things like transcription, you'll see a series of dark bands which represent different extension products. So at the very bottom, you should always have your starting primer at time 0, for example, whether it be a control or some sort of DNA adduct. But most of the time, you'll see that these controls fully extend. In this example, I hypothetically said this is a 12-mer. It extended all the way at 30 minutes to its 18-mer. But for something like a DNA adduct, what happens is because you change the structure of DNA, or sometimes the adduct is so bulky that DNA polymerases can't necessarily fit around the DNA polymerase and incoming deoxynucleotide phosphate or ribonucleotide triphosphate complex. And what ends up happening is that you have a series of shorter extended products, and sometimes you never even see a fully extended product. These assays are also really important whenever you're trying to do studies with DNA replication or transcription, and when you talk about things such as viruses, you're trying to test out a series of inhibitors, you can run these kinds of assays in order to see that your inhibitor is actually working. The other two kinds of experiments that you can do with this primer extension assay are two very similarly related assays, which are the single nucleotide incorporation assays and the kinetics assays. The single nucleotide incorporation assay uses only one type of dNTP or NTP, and you see its insertion across a DNA adduct site or its addition to other sites along that strand. You also have the kinetics assays, which you also only see how the additions of one dNTP at different concentrations changes over time in order to know how efficiently, if it's a mismatch for example, how efficiently it actually gets inserted to the opposite strand. So one of the next things you have to do for this assay is of course set up your gel. These gels, as you can see from this glass, are quite long, and that means they're quite thin. So they are very fragile, and they also take quite a while. You should do this step as early as you can in the day whenever you're setting up this assay. Sometimes you'll have the best of luck and your gel will come out cleanly and look great your first time attempting to set it. However, sometimes you may have a bad day and you might have to try 3 or 4 times until the gel looks nice and sets. So make sure that you've effectively cleaned these glass plates as well as the plastic spacers that go into these glass plates before you ever make your gel. What I do is that I spray these with water, make sure that I can get all the hard little pieces of gel that might have been there from a previous run scratched off of this glass slide, and then I go with ethanol, and it works twofold. It works to make some of the glass or some of the gel pieces that were there before a little bit easier to scratch off if you couldn't get them with water, because it dries them up, and it also dries up the water much quicker. The next thing you have to do after you clean the glass plates is that you should siliconize one of the glass plates. This is really important because if you do not siliconize the glass plate, or both of them, you can have tearing of your gel. What happens is that whenever you take these gels off of the apparatus, they start to cool down quite quickly. And when they cool down, they start to stick to the glass. And whenever you try to pop it open, you might get a tear. And of course, then that's just an unlucky day. And we want to prevent that as much as possible. So the best way to do this is by siliconizing it, which adds a hydrophobic layer to the glass and makes it really easy for your gel not to stick onto the glass and also pop it off at the same time. So to do this, whenever your glass is nice and clean, you should go to a hood. Once you have placed the glass plate in the hood, the next thing you want to do is you want to get a siliconizing reagent. The siliconizing reagent that I like to use is this Sigma coat that you can buy from Sigma Aldrich. And so what you do is you uncap it, and then ever so slowly pour it out into the middle of the glass plate. You only need a very tiny amount. And then pick it up a little bit and roll around the liquid on all sides. You are done once you have your siliconizing reagent in all parts of the plate, which will be important in preventing the sticking of the gel. This does take a little bit of practice to get done right. If you feel like you need more siliconizing reagent, feel free to use it. According to the manufacturer, you can use any excess siliconizing reagent. You can put it back in the bottle for reuse. I, however, don't do that. I like to just use a little bit of it and call it a day, just to make sure that the next time I use a siliconizing reagent that's in that bottle, it's actually fresh and not made up of somewhat used siliconizing reagent. Now the siliconizing reagent is almost instantaneous, and you can just start to wash it off as soon as you have placed the siliconizing reagent all over your glass plate. However, I like to leave it in the hood and let it dry up a little bit. It usually takes only about 7 to 10 minutes. The heptane solution that it's stored in goes off nicely, and the little gaseous hydrochloric acid that's produced can get off into the vents of the hood. But once this step is done, all you have to do is wash it really well with water, and you'll be able to tell that the siliconizing is done because the glass feels a little bit oily. It feels like you can actually run your fingers over it very smoothly. The other way is, of course, when you start washing it with a lot of water, you'll see that your water, instead of trickling down, it actually bubbles up. The gel piece that I use is always the longer piece that doesn't have that U cut out here at the top. As you can see, there's this little strip that's actually a temperature strip. I always make sure that's facing down. I use a pair of round bottom flask holders in order to nicely even out the glass plate. So what you have to do is you have to place these spacers. The long ones go on the sides, and then there's a shorter one that goes at the bottom. The short one is actually quite important. As you can see, this short spacer actually doesn't fit the whole length of the gel. And the reason for that is to allow some of the SDS page material to go in between it. So whenever some gel forms down here, you can still have a current, and that allows for your gel not to get overbearingly hot, because if your gel gets way too hot while you're running it, anywhere above 65 degrees, there's a potential that the plate could start cracking, which if it does, then your gel plate is probably no longer good. To prevent that, this smaller piece helps to form some of the gel piece down here. Of course, you could also ask the question, then why do we put the spacer there at all? Well, we put the spacer there not overcomplicate the gel, because then it could easily leak out, or it's easier to make a smaller gel than it is to make this much longer gel. Once you're done, you get your glass piece. Make sure that the siliconizing face is actually placed on top of these spacers. Do whatever you have to do in order to remember. Sometimes I use a Sharpie on the opposite side where it's not siliconized in order to denote that that's the siliconized side. But in this case, I know that this is the right side because this glass piece actually happens to have a small crack at the right corner, and so I know this side is not the side that I've siliconized. So once you put the glass slides on top of each other, sometimes these spacers tend to move. So if you have to move them ever so slightly, you can. But I'm just trying to make sure that the spacers are as even as possible. And you can see that there's some space right here at the bottom so that some gel material can go here, form, and be able to pull a nice and even current across the gel so it doesn't overheat. The next step is that you want to use these clamps. So these clamps can be used right here, somewhere a quarter of the way, both on the top and the bottom of these gel plates. And this will help hold those spacers in place and the whole apparatus while you construct your gel. You just want to make sure that they don't move too much. Sometimes when you have this gel plate on top that's siliconized, it tends to be a little bit more slippery because of that hydrophobic surface that it has now. But basically, as long as everything is nice and even, you're good to go. The important part is that this bottom edge down here is as even as possible, because the next step is that we're gonna have to tape it. We have to use this gel tape here in order to effectively seal the bottom, because as you can imagine, when you're pouring the gel, if this is open at all, it will pour out. So the best way that you can do is to kind of get a long piece of tape. It's a little bit longer than the bottom side. Right here. Get that piece of tape and do your best to even it out with the bottom. And then run your finger over the bottom to make sure that this tape has basically sealed here. And do you see these bubbles right here? So try to push those out. Make sure that this tape on the bottom is even. If you have to push out the tape a little bit and try to redo it, you can. This tape is actually quite easy to get off. As you can see, just like so. And then on the edges, do the same thing. Run your finger along right there. Once the edges are nice and sealed, you can then start to push on top. Make sure that you start with one end and rub from the bottom towards the top, like so. Make sure you have as very small bubbles as possible. You don't want those bubbles. Bubbles are your enemy. But again, as long as that bottom layer is nicely sealed, doesn't matter too much about the bottom. Make sure you do the same with the back. The back is a little harder to see, but of course, if you angle your head, you can see the back and how it's coming and how that tape is also coming along. All right. You can, of course, use a second layer of tape if you so wish, in order to make sure that the bottom is effectively sealed. I have had personal experience that whenever the gel really leaks, when it really starts to leak, it leaks out of these corners. So I actually like to instead tape up the corners a little bit better, but this will be done a few times. The other thing is that you don't necessarily have to tape up the corners. And of course, if you do need some help getting that tape off, you can get a tiny little razor blade to effectively get one of the corners. But as long as you seal the bottom quite well, you don't need to actually seal the sides of the plate. You can, and in my experience, you should. If anything leaks out of the corners, can leak out of the sides. So you should just tape everything, minus the top. Okay, that's one corner. Make sure there's no bubbles. There you go. Right. And that's the second corner. All right. Now, just to make sure, I'm going to also tape the sides of this plate. So again, get a piece of tape, a little bit longer than the actual length of the plate itself. If it's a little too long, you can put some of the excess down here at the corner it doesn't hurt to have more tape at that corner. And what you want to do is that you want to remove these clamps. Do a similar procedure in which you get it to the length and you start to slowly roll your fingers across. Make sure that the plates are even. So that there's no bubbles. Importantly, here where the first seal is at. You see there's a little overhang, but that's okay. And go at the top. Make sure you don't have as many bubbles there at the top as well. If you do, just try to push them out with your thumb as best as you can. The most important seal is the seals on the sides. As long as those don't have bubbles, you should be pretty confident that your gel solution should not come out. Okay. Then after putting it on the tape, make sure to put the clamps back on just to help with the mobilization. And then you rinse and repeat on the other side now. All right, how's this going, Broccoli? Yep. Of course. Oh, of course. All right, and now you're done. You have effectively taped the sides really well. If you have any corners that you might be a little worried about, for example, again, the corners that I would worry about are down here on the bottom of the gel. You can retape those as much as you'd like. I wouldn't go over 3 layers, of course, because I feel like that's not only wasting your time a little bit, but that's also will make some of this tape harder to take off, because eventually once you lay down the gel, let it set, you do have to take the pieces of tape off. Now, since most of the time you'll be doing this gel alone, the next thing that we gotta do is actually cast the gel. And so to do that, I usually take some of these microcentrifuge trays, place them right underneath the gel apparatus, and I try to balance it out, put it at an angle. All right, so this will make it easier for you to actually pour your gel solution down the gel. So now it's actually time to make the gel solution. So for the gel solution, I like to use a 250 Erlenmeyer flask. I measure out at least 100 mils of 20% pH solution. And then that 20% pH solution, we're gonna mix it with this salt called ammonium persulfate. It will dissolve eventually over time. You'll just have to shake your flask by hand. It should dissolve quite readily. And then finally, whenever you're ready to lay down the gel, the next important piece is this TMED. This TMED is one of the radical initiators, so it will start the polymerization process. It'll be pretty quick, so you'll want to move quickly, but not all over the place. Once you add that TMED, you should have a syringe in hand. This syringe has a little bit of tubing and this pipette tip that has been cut off with a razor, so the inner diameter is actually quite a bit larger. So the reason to do that is that it effectively helps you lay down the gel quicker. Whenever I have the gel solution here, I'll put the syringe and now pull back on the syringe in order to take the solution out of the Erlenmeyer flask. I'll actually be putting the Erlenmeyer flask at an angle, probably inside this tape. And that's to help me get all of it in the syringe as fast as possible. I'll have to tap it to get some of the bubbles out, but as soon as it's nice and even in there and in the tubing, I'll have to just start laying it down on the gel plate. Okay, so here we go. So here's 100 mils of a 20% pH solution into this 250 Erlenmeyer. Here I have about 85 milligrams ammonium persulfate weighed out. Pour it into the Erlenmeyer and swirl. Again, this is a salt. It won't dissolve readily or quickly, so you want to, what you want to do is, as you mix it, you hold it up, the flask up to your eyes, see if the salt has effectively been dissolved. If it hasn't, keep stirring manually. It's almost there. This usually doesn't take more than a minute, but again, you want to be quick. There's one last little granule, and that's it right there. Next, I add 85 microliters of this TMED solution. If for any reason this TMED solution causes too quick of a polymerization, you can use a different amount. Maybe try 5 microliters less. We input the solution, we swirl it a little bit, and then we start. Of course, as you draw it in, you will create some bubbles. That's fine. Just try to do this as quickly as you possibly can. Put it at an angle if you must. Oh. So it looks good. No bubbles in the line. Good. Then start. The gel will polymerize quite quickly, so you want to get an even Don't worry about any of the bubbles that form. They should come out right at the top. Remember, this gel is only going to be ran halfway through, so if there's any large bubbles at the bottom, don't worry too much about them. If you start seeing bubbles at the top, however, this is where you're going to try to help out the system by quickly just taking it up just a second, just to let those bubbles come. Tap the glass, then quickly lay the gel down and insert the comb. And it's done. So the next thing you want to do after you put in that comb, visually inspect that there's no bubbles anywhere in the area where you want to do your experiment. So I'm gonna run my gel halfway through, and so as I'm visually inspecting it, I see no bubbles here. I see no bubbles in the wells, which is fantastic. I can tell that my gel is going to be set nicely. This will take about an hour max. Now that the gel is properly set, you can do other things while this is setting. Of course, you can get your samples ready to go for the actual primer extension studies, which means that you have to gather your materials of radiolabeled DNA, the polymerase of interest, which in our lab, we use polymerase ETA, which is a translesion synthesis DNA polymerase, as well as a mixture of the DNTPs in 50 micromolars of each. So we'll gather those materials together, ready to go for the experiment that will occur using this gel. If you're wondering if your gel has solidified, usually you'll have a bit of solution still left here in your flask. As you can see, we have quite a bit, still about 10 mils. You'll probably also have another bit here found in your syringe. Make sure that once these have polymerized, that means that your gel should also be completely polymerized. So once you have done setting up your gel, aside from getting your samples ready that you will run, the other thing that I recommend to do is also to clean up your workspace. Setting up these radioactive gel assays can get really hectic really quickly, and so you will have all sorts of things laying around. So try to clean up your space as much as possible in between steps that have some time in between. So for example, over here, the only thing that I have left next to my gel is the Erlenmeyer flask that still has some of the page gel solution, just to watch for it to polymerize, because once it has polymerized and the gel's done, that syringe, I'm just keeping it over there to remind myself that I need to clean it out. The best way to do so is, if with this syringe, if you're to take off the tubing, you can just get a pipette tip, stab at the opening once you've removed this tube in order to draw in some air to take off that little piece of gel that will form in the syringe. And then you can fill the syringe with water and do your best to push out this piece of gel that's forming within the inside of the tubing. The other things that you should do is to put up the reagents that were used to make the gel, prepare the gel apparatus. So things like the Sigma coat reagent, you should put that back at least in a 4-degree fridge, as well as the T-Mit and ammonium persulfate. Those guys should always stay cold, and so also put them in a 4-degree fridge. Let's talk about a few things before we even start this experiment, all right? So, proper lab safety. What you'll need is to always be wearing your lab coat. No matter what you do, always be wearing this lab coat. Make sure that you also put on 2 pairs of gloves. Now, why put on 2 pairs of gloves? Well, just in case if you have any sort of radioactive material that does compromise one of these gloves, you can easily take that pair off and throw it away, and then replace it with a second pair of gloves. You always want to have 2 pairs of gloves on your hands at all times in this room. And then the last thing that I think is a good idea to always have is to have your Geiger counter on at all times. Now, the Geiger counter really is only to make sure whenever you get out of the room to make sure that you're not carrying any sort of radioactive materials anywhere on your lab coat or under your shoes or on your hands. We'll talk more about that in a little bit. But I like to have it on at all times during the experiment, just in case I do contaminate my hands. I don't all of a sudden contaminate the whole Geiger counter apparatus, and then I'm touching things and I'm spraying it everywhere. So it's always just a good thing to have in mind. So the primer extension assay. So what do we do today? It's a 2-day process. A 3rd day if you include the imaging. But for the first day, what you want to do is you want to radio label your primer that you're going to use and then anneal it to its template. Ideally, after you've set it up, you can use it a few hours after annealing, but in my experience, it's best to just prepare it and leave it overnight and come the next day and use your annealed template. So what I like to do before I even come into the room is to prepare the samples. These samples are 3 different things in my case. So the first one is a sample that contains T4P and K within a T4P and K buffer with the amount of DNA primer that I want to use. In this case, let's say I have about 500 picomoles. The reaction volume of this is about 20 microliters in total. The one thing that I only do here in this room is whenever I get my primer, that's always on ice, by the way. Keep your stuff on ice until you have to take it off of ice and add stuff. The one thing that I add here in this radioactive room is, of course, the radioactive ATP, or gamma P32 ATP, whatever you want to call it. What happens is when you use T4PNK enzyme, which stands for polynucleotide kinase, what it does is that it takes that gamma phosphate, which is the terminal phosphate on ATP, and it transfers it over to the 5' hydroxy position of your DNA primer. And by doing so, it effectively radiolabels your DNA. What we do here is in this 20 microliter volume, we add 1 microliter of radioactive ATP, which is about 24 microcurie. This radioactive ATP is found within this freezer here. It comes in this blue container, and we purchase ours from Perkman Elmer. Make sure to note the lot as well as the expiration date and the date that you actually got this, which should be on the label. This will tell you how many half-lives this radioactive ATP has been through. The half-life of radioactive ATP is about 2 weeks. So to get the nicest data you can, try to use it within the first 2 weeks of purchase. You can use it up to a month after purchase, but after that, I would not recommend it. The gels that I've done that have used past month-old radioactive ATP look awful. So you'll want to get this whenever you absolutely are ready to do these radioactive assays. You'll want to let this come to room temperature. It takes a few minutes for it to defrost, anywhere from about 10 to 15. But the actual containing the radioactive ATP is found within this blue container. From there, all you have to do is take 1 microliter again of this radioactive ATP and introduce it to your primer that you'll want to radiolabel that contains T4PNK and T4PNK buffer. T4PNK and the T4PNK buffer can be purchased from New England Biolabs, which is what our lab has done in the past. But once you've added your radioactive ATP, again 1 microliter, which is 25 microcurie, into your solution, you should have 20 microliters overall volume in this solution that will radiolabel your primer. You then put that solution on this heat block that's preheated at 37 degrees, and you allow this reaction to occur for 1 hour. So the reaction that's going on right there is being catalyzed by T4PNK enzyme, or T4 polynucleotide kinase. So this enzyme catalyzes the transfer of the terminal or gamma phosphate of ATP onto the 5' end of your DNA primer in this case. When we use radioactive ATP, which has a P32 labeled phosphate at the terminal end, what happens is that you're transferring that radioactive phosphate group onto the 5' hydroxyl group of the primer strand. After you're done using your radioactive ATP, make sure you put it right back in its blue container and right back in the freezer. When you're working with radioactive material, you only want to keep it out as long as you possibly need to have it out. So please, right after use, put it back. The next thing, once the hour has passed, what we're going to do is we're going to remove this radioactive primer and move it from 37 degrees over to this heat block that has been heated up to 67 degrees. The reason we are treating this reaction at 67 degrees, it's to heat inactivate the enzyme that's catalyzing the addition of this radioactive phosphorus onto the DNA primer. We're going to do it for 10 minutes. We don't need it anymore, and so this will help us easily kill the reaction. So after having this on the block for about 10 to 15 minutes, you have effectively inactivated the enzyme. So the next thing to do is to introduce it into one of these little G25 Illustra microspin columns. So what are these columns? They have a packet material that effectively captures any radiolabeled ATP that hasn't reacted. However, at the same time, it allows you to elute your radiolabeled primer, which is great because that's what you want. In order for these to work, what you have to do is that you have to snap off this triangular end off of these columns and then put it on an Eppendorf tube. You can also untwist the cap ever so slightly, just a quarter of a turn, just to allow some of the air to go into the column. You'll want to balance it out with another column that's not opened, and then centrifuge it down for about a minute or two to remove any of the storage buffer that's found within the column, to then take off the column off the microcentrifuge, take off the storage buffer, What you would do then is replace the Eppendorf that had the storage buffer in with an Eppendorf that should be labeled P32 primer, and then have any sort of other labels that will denote it as the primer that you're radio labeling, as well as the date and other important things to be able to identify it. Once you have introduced your 20 microliter aliquot sample onto the column itself, you then put it back onto the centrifuge with a balance, and then you centrifuge it down for another minute or two until all your sample has come out. From there, once you remove the balance and then your sample, you can then dispose of this column into radioactive waste. You can put it into this beaker, which is marked for radioactive waste. This beaker is something that we use here just so whenever we have time and once it's full, we can actually throw it into the physical radioactive trash. This is just handy for us to have because, of course, it would be a really big hassle for us to keep constantly opening up the radioactive materials waste bin, when we can have something small like this and just keep it around to just throw things away. Once you have radioactive material in hand, you can then put this primer on its own back into the freezer where the ATP is found. Again, label it really well as what the primer is, when it was made, in order for you not to mix it up with potentially older samples you could have in this fridge. Of course, we're not done yet. The next step is to anneal the primer onto the template strand. But for example, if you had something to do in between those times, you can put this away and store it in the freezer. It'll wait for you there until you come back. So now the gel has set. It's been about an hour. To take it apart, to set it up in the actual gel box, you're gonna have to take off the clamps from the sides, and then the tape on the sides as well. So you should start with the sides before the bottom, because tape on the sides is on top of the tape on the bottom. This can be a little tricky, so just do your best. Try to leave as little tape residue on the plates as possible. Sometimes it does interfere with how current runs, but if you see a little bit of tape, like that little speck right there, it should be okay. That's one piece of tape. Flip it over. Repeat again. Okay, and as you can see, this last piece of tape really stuck onto the bottom. So we're going to use it to help us take off the bottom piece of tape. And there you go. Now you have your gel out of the tape. You can see that the gel is perfectly inside there. There is some extra pieces hanging out, but that's perfectly fine. So the next thing we're gonna do is move this over to the gel box apparatus. So now that I have my gel ready to go, this U-shaped piece of glass should actually be facing the back of the apparatus. So I'm going to give this a flip. And you know you have the right side of the gel whenever you see this indicator. This indicator is here just to make sure that you know how hot your gel pieces are, which is quite important. There is such a thing as overheating the gel box. If you do, you could potentially crack your glass. If for any reason there is some sort of buildup on the back glass plate, make sure you clean it off really well. You can just do this by using water and then using towels in order to dry it and effectively take off that grime. See, for example, here in this case, we actually have some buildup here on the front of the gel. We can take that off by, again, just using a little water and taking off those gel pieces. Now, before I get to do that though, I'm going to tighten the gel here. So these flaps on the side of the gel hold it in. All you have to do is screw them in, and do so to keep them nice and even. Try not to apply too much pressure to one side at a time. Try to apply more pressure on one side versus the other. Now, if you're curious if you have tightened it enough, what I use to make sure is whenever you actually put in the running buffer. The way this system works is that if you have made this gel tight enough in the gel box, whenever you introduce your running buffer in the top compartment, there shouldn't be any leakages in the back side of the gel. If you do see some leakages, this means you might have to tighten the top portions, the middle portions, and the bottom portions a little bit more again in order to get a nice tight seal. Because of course, you don't want your running buffer actually going through the back, because this back portion of the aluminum casing of this gel box gets quite hot, and you might start to hear crackling and other noises, and it could unevenly heat the gel, and it could cause damage to the glass pieces themselves. So make sure that there's a nice tight seal in the top. So let's test to see if we've made it tight enough. Our running buffer is this 1X TBE. TBE standing for Tris, Borate, and EDTA running buffer. This we usually make in the lab in a 10X concentration, so we just get a portion of it, let's say 100 mils, will be diluted by 900 mils in order to get a 1X TBE. This instrument requires about 1 liter max, and so here I have about a little over 1 liter just to make sure that if I need any or if I spill some, there's still enough to run the experiment. So we're gonna pour it at the top. All right, that should be enough. As long as you have enough TBE over the wells, that's great. That's all you need. And as you can see, not only is there enough TBE above the wells, but there's also no leakage of TBE into the back part of the aluminum casing. So in other words, everywhere along this gel, it's perfectly tight, so we don't need to tighten it anymore. The next portion is to actually get the 1X TBE into the bottom chamber. You can do this a variety of ways. If you didn't pre-fill it like I did in this case, you can always try your best to scooch the bottom powder a little forward, and I used the front plate in order to pour it in there. So as you can see, there's only a little bit of it coming out. There is a fill line on this instrument. All right, and it's at the fill line. Any little bit that fell, you can just soak it up with a cam wipe and it should be fine. So now, before we run the apparatus and close it, there's a couple of other things we have to do. So one of them is to take off the gel comb. Previously, we used to have a gel box where we didn't have to take off the well comb until the gel was hot, and that made the comb a lot easier. In this case, the gel's wet, but we have to remove the comb because when we try to close it, as you can see, there's still a gap, and we don't want to force that comb to bend in there. And the reasons for that is you could destroy your wells. I've done that before because I was wondering what would happen, and it's not a good time. So I would recommend for you to take off the comb before you even get any of this started. So the best way that I'm gonna try is that I'm gonna slowly peel back here. As you can see, as I push the comb, you can see some of the buffer that I added is slowly filling into the front and might get into the wells itself. That kind of helps me loosen up some of the comb. Next, we're going to slowly lift it out here from one side, then the other as well. Again, pull back a little bit. There we go. Okay. Using my other hand over here, just to help pull a little bit of it up. And there we go. Now we got the comb out of the gel. And so now we've exposed our wells. Our wells will be fine. There's a bunch of the wells are lopsided currently, but they'll be fine. They'll fix up as soon as you start running the gel. So now it's time to get the gel started. So we do this by first closing the top, then sliding it. If you hear that click right there, that means that you have effectively closed the top of the gel. Next thing we're gonna do is we're gonna actually plug in the power generator. We're gonna flip the switch to on. Currently it's not running just yet. We have to put the negative and the positive end into the instrument. Make sure that you put it in the right color and the right spot. So black with black, red with red. That. Here you can already see that it has a timer for 5 hours. This is typically because of the fact that we run the gel for about 30 minutes to an hour to preheat it. During that time, we're running one of our assays, and then we stop it, and it usually takes about another 3 hours max to run the gel. Now that we've plugged in our gel box into the power generator, what we need to do is set up how many volts to run as well as the time. Usually automatically what you see here on the power generator is that it runs for about 5 hours. This is because of the fact that we run our gel for about 30 minutes to an hour to preheat it. During this time, we're usually running one of our assays. Then after we're done, we turn off the generator for a split second just to add our samples. And then it runs for an additional 3 to 4 hours depending on what kind of voltage you're running across the plate. So typically for this gel box, we run about 1600 to 1800 volts max. So samples will be ran through the gel in about 2.5 to 3 hours. They'll separate nicely in order for us to get phosphor imaging done. So what we want to do is press the on button right here. And as soon as you do that, you can see that it's already starting to, the voltage is starting to go up. As soon as it stops, you can then start to change the voltage. If you turn it clockwise, of course, it will increase the voltage. If you turn it counterclockwise, it will decrease the voltage. Right now, it's at 1850. Let's decrease it just a little bit back, closer to 1800. And that's good enough. So once you hear those beeping noises, it will only run it at the voltage that you set it at. So 1800 volts is probably the maximum amount that you can do for this gel box. You could try to run it at 2000 volts, but at 2000 volts, the gel might be getting a little too hot. And so depending on what kind of experiments you're running, that it might get so hot that your DNA can begin to degrade, your RNA, which is a little bit more scary, or you might heat the glass plates too much that they actually are so hot to the touch that they will then crack, which is the worst case scenario. So I recommend, from personal experience, 1600 to 1800 volts max is what you should be running this gel box at. The other thing about running the gel box hot is that it's better for your samples. So you could, in theory, just run your samples on the gel cold, but two things tend to happen. If you run your samples cold, the separation may not be as good as if it were hot. Sometimes it streaks and it looks just really bad when you phosphor image it. The second thing is that if you run your gel cold, your samples also tend to take a lot longer to develop. So while running my samples on a hot gel might take 2.5 to 3 hours at this voltage, it may actually take a lot longer to run that same amount of samples on a cold gel, anywhere from 4 to 5. And so since this assay is already quite long, you might as well just save yourself a couple of hours. So now that we've come back and took your sample out of the freezer, so now you have your radio-labeled primer here. Now, whenever you do any sort of work that has any sort of radioactive material, make sure you're always behind this blocking screen. So as you can see, my body, my face, everything is behind this screen right here, but everything else, all my samples, are where I can work from behind this blocking screen. This is just for safety concerns. So now that I have my radio-labeled primer here, all nice and thawed out, the next thing that I want to do is to anneal that primer onto a template strand. So in this case, I have two other samples here that contain two different kinds of templates where my primer is going to attach. In this case, one of the templates is just a control DNA, and the other template, for example, is a DPC, so it contains a DNA adduct of some sort. For DNA annealing, what we do here in our lab is that we usually take one equivalent of our radio-labeled primer to two equivalents of our template strand. So the reason we do this is because by doing these kinds of ratios, we ensure that all the primer gets annealed onto its template strand. The rest of the template strand that doesn't have a primer won't even show up on the radiolabeled gels. Because again, since we're doing phosphor imaging, the only thing that should show up on the gels is a primer. Also, this kind of entails the same go around for the primer. If the primer isn't annealed to anything, it won't run on the gel other than staying at your time 0 whenever you collect that point during your experiment. So what you have to do in order to anneal your DNA, take out 1 equivalent of your primer, then quickly mix it with 2 equivalents of DNA, and then take that pipette tip and put it over there in the radioactive waste beaker. And then you repeat this again for as many samples as you need to prepare annealed product for. So now we're going to move both of these samples that have both template and primer strand DNA onto this preheated heat block at 95 degrees C's. So what we're gonna do is we're gonna heat them up. We're gonna let them sit there for 5 minutes. Afterwards, we're going to switch off the heat block and let them cool down to room temperature. So basically what you're doing here is that you're allowing these DNAs to float into solution, and as the solution cools, they will in space meet each other and anneal and make primer template duplexes, which is what you need in order to run these kinds of experiments, as polymerases recognize this double-stranded DNA. Whenever you anneal template DNA onto primer DNA, you can leave it overnight, which is what I would recommend, or if you're really in a hurry and you gotta do this experiment, you can let them sit for about 2.5 to 3 hours. From there, you can use these primer template DNA duplexes on the same day to run your experiments. After your overnight incubation, or your 2.5 to 3 hours of letting the DNA, primer, and template strands come together and anneal, the next thing to do is to actually run your polymerase assay. So here's a few things you need to know. Most of the components of these reaction vessels will be done outside of the radioactive room. The only time you have to actually take your samples back here is when you're ready to start adding radioactive DNA. While you're outside, these little reaction vessels here are about 30 microliters in total volume after you've added all the components you need to add. This contains currently just the reaction buffer for the enzyme that you'll need to run the reaction in. The reaction buffer typically contains salts that are important for the activity of the enzyme, such as magnesium chloride, since polymerases need this magnesium in order to coordinate any sort of deoxynucleotide triphosphate that's coming in order to catalyze the reaction. Here in the room, we're going to add 2 things to those reaction vessels. I like to keep everything on ice until I have to add my radioactive DNA. So here is a portion of the enzyme that we're going to use in today's experiment, which is called ETA. And then I also have a master mix of all 4 different DNTPs, such as DATP, DGTP, DCTP, and DTTP. So a master mix of all those DNTPs at a concentration of 50 micromolar each, which is what you'll use to initiate the reactions. Here in this ice bucket, we have 8 different samples. These different samples, again, have currently reaction buffer where the enzyme will work best. Here we have an aliquot on the enzyme, and then here we have DNTPs. So, what we're going to do is that we're going to put aliquots of the enzyme into each of these reaction vessels. From there, to initiate the reaction, we need to incorporate DNTPs into each one of these vessels. This is the very last thing we want to do because, of course, once you start the reaction, you're committing to getting samples at particular time points for this reaction. You will run this reaction at 37 degrees Celsius in order for the polymerase to work efficiently and to catalyze the addition of these DNTPs onto your primer. And so you should see addition products over time. What we first want to do to these reaction vessels is add the polymerase. We will want to keep these reaction vessels on ice until you add polymerase to each one of them. Once we do, we're going to transfer these polymerases over here behind our blocking screen onto where our set of samples will be collected, as well as where the radiolabeled DNA primer template strands are. So first, going to get an aliquot of your enzyme. typically 1 microliter, which is about a picomole of enzyme for this particular assay, and mix it well. And then keep doing that for every reaction vessel. Of course, replace the tip. So after adding polymerase to all of these different samples, I now put them behind the blocking screen. And if you're wondering why, it's because we're about to incorporate DNA primer templates that are radioactively labeled, and so you would want to do this behind the blocking screen. However, whenever we put these radioactive DNAs into these samples are now going to stay here, not back on ice. Because if you think about it, you can accidentally pass radioactive material into the ice, and then you've contaminated the ice, and then it just starts a whole thing of where the ice went, and then you have contamination all over the place. And then second, of course, once you add your DNA, the next thing you want to do is incubate it on the heat block anyways, and so this makes it convenient from once you've added your DNA, you put it on the heat block, and you're set to go. A few things before we actually also start to put things on the heat block, we should probably talk about the sample system that we have here. For these samples, I'm going to be taking different time points from 0 minutes, 5 minutes, 15, and 30. That's why, as you see here, for samples 1 through 8, I also have respective 4 samples for each of this reaction vessel. So for example, the way that I number my things is for reaction vessel 1, I have time point 1-1, 1-2, 1-3, and 1-4. In my mind, 1-1 is your time 0, 1-2 is my 5 minute, 1-3 is my 15 minute, and 1-4 is my 30 minute. And the same numbering system goes for each other reaction vessel. So for example, if I picked reaction vessel 2, I have sampled time point 2-1, 2-2, 2-3, and 2-4, and so on and so forth. Again, since I'm taking different time points here, at least for me, it helps me to have a timesheet as to when I'm going to be picking out these time points. For these different time points, I've set up a way as to how to efficiently get to each time point and not miss one. Because this is, of course, critically important. You want to get your sample into a quencher in order to collect that time point. So the way that I do this, especially since I only have 8 samples, is I do 30-second intervals. This timesheet helps me out in able to figure out when to take samples into a respective sample vessel here, where I can just quench the reaction. What I do is that I do sample acquisition every 30 seconds. For example, at time 30 seconds, I add 1. What that means to me is I get an aliquot of DNTPs with a 50 micromolar concentration and add it to reaction vessel 1. And then I put it on the heat block and let it incubate at 37 degrees Celsius. And since I'm giving myself 30 seconds to do each of these things, then that means at minute 1, I'm adding DNTPs to reaction vessel 2. At minute 1:30, I'm adding it to 3. At minute 2, I'm adding it to 4, and so on and so forth, up until I get to the last reaction vessel, which is 8 at 4 minutes. So then, if you do the math, since I started the reaction in reaction vessel 1 at 30 seconds, and I'm trying to take a 5-minute aliquot, that means at 5 minutes and 30 seconds, I'm going to get my first aliquot of reaction vessel 1 from the heat block and put it into one of my reaction vessels in order to quench that. Usually we get a 5 microliter sample from the reaction vessel and quench it with 4 microliters of a 95% formamide solution in order to effectively quench and collect that time point. And I will keep repeating the same idea of 30-second intervals between each sample take up until I'm done the experiment. So in other words, since I'm only taking 0, 5 minute, 15 minute, and 30 minute time points, I should be done by 34 minutes. Here where it says comment and here where it says time, at time 30 seconds, I add to 1. What I mean there is that I'm going to add my mixture of DNTPs into reaction vessel 1, and then once I do that, I put it on the heat block. And then at minute 1, I do the same for 2. At minute 1:30, I do the same for 3. At 2 minutes, I do that for 4, and so on and so forth. Up until I get to minute 4, which is the addition of my last set of DNTPs into reaction vessel 8 and put it on the heat block. So now we've added our radio-labeled DNA primer templates into each of these reaction vessels. So currently at this point, what we have here is a reaction vessel that contains polymerase and radio-labeled primer DNA duplex. So to initiate the reactions, you need to add DNTPs. A very important thing that you need to do before you do any of that, however, is to take your first time point. So when you take your 0 time point, what you'll want to do is take 4 microliters of the reaction vessel and put those 4 microliters into its respective ×0 time point vessel. Here, for example, I have reaction vessel 3. I'm going to take 4 microliters, and then I get 3,1, which is time 0 for me for my reaction vessel 3. Mix it up really well. Now I have an aliquot for time 0 for reaction vessel 3. And basically, I just keep redoing this for every different reaction vessel. And again, for me, everything with a subscript 1 is time 0. So for the next part, we're going to add 50 micromolar solution of DNTPs into these reaction vessels. And so this is where the actual experiment is going to start. What I like to do here is just have my set of DNTPs here on ice, in this ice bucket. And I will retrieve the DNTP every time I need to add DNTPs into the reaction vessel. So now it's time to actually start the experiments. With the time 0 time points collected, now all you have to do is to add a DNTP mixture of 50 micromole concentration into each of these reaction vessels to get it started. Now, I like to keep these DNTPs on my ice until I have to add them to the reaction vessel. Once these DNTPs are in the reaction vessel, now you have polymerase, DNA, and DNTPs, which means that these reactions can now occur. You will incubate them at 37 degrees Celsius in order for the reaction to proceed efficiently. Now this is where your choice of what you want to do for the timer comes in. We here in the lab have a timer that you can start as a stopwatch. You can keep, if you have a watch on your wrist, you could always start a timer on your wrist. You just have to know your system. For me personally, I like having a watch on my wrist in order so I can look every few seconds what particular time I'm at. And then once I know 30 seconds have passed, I can add my first additional DNTP into reaction vessel 1, incubate it, and another 30 seconds have passed, add more DNTPs, but this time to reaction vessel 2, incubate it, and so on and so forth. So now let's get this experiment started. So like I said, I like to use a stopwatch on my wrist. So here we go. All right. I usually like to wait about 15 to 20 seconds prior to adding the DNTP or taking an aliquot from the reaction vessel. So DNTPs, I got it. Reaction vessel, I open it. I position my tip right above it, and now we fit 30 seconds. Mix it. It goes on the heating block. And then it's just rinse and repeat. So another pipette tip ready to go. I'm currently at 42 seconds. I'm going to collect a fresh portion of DNTPs. Here's my aliquot. Cap it. Return it. I'm at 53 seconds. Position my DNTPs right above, and we're at 1 minute. Mix them, put them on the heat block. And then this is what I'll keep doing until I add DNTPs to every single reaction vessel. Now, as you can see, all reactions are on the heat block. I did 30-second intervals, so whenever I added DNTPs, I added it to reaction vessel 1 at 30 seconds, then at a minute, minute 30, 2 minutes, 2 minutes and 30 seconds, and so on and so forth until I filled all of these reaction vessels. Now, my first reaction vessel time point will be at 5 minutes and 30 seconds, because if I'm taking a 5-minute time point, if I started this reaction vessel at 30 seconds, 5 minutes later would be 5 minutes and 30 seconds. What I'll do then is I'll get my micropipetter, set it to 4 microliters, and get ready to get my first aliquot from reaction vessel 1. So we're currently at 5 minutes, and so I'll let this go for a few more seconds, about 15. So we're nearing the time now. Gonna get reaction vessel 1. Gonna get a 4 microliter aliquot. Then I get the sample that corresponds to that time point. So 1, 2, is 5 minutes for me. Now, I'm gonna place my sample directly above it until I hit the time point, which is now. And I add my sample in here, briefly stir it, and make sure that almost all of it is gone out of my pipette tip in order so I can have maximum sample size. And then, since I'm repeating it every 30 seconds, get my sample from reaction vessel 2, go to my 5-minute time point for reaction vessel 2, and add. Close, and get rid of my pipette tip. And get ready for the next time point to get, which is from 3. And this will keep repeating itself over and over again, 5 minutes, then at 15 minutes, then at 30 minutes. So now we are done with the assay, and we've collected our samples here. Next, we're going to heat them up for about 5 minutes. Sometimes with this percent of formamide, they can get a little sticky, especially because you have some protein in there. And by heating it up, it really makes everything a lot easier to just get out of these microcentrifuge tubes, and then also easily put it on your gel. So again, we're going to heat these samples up for about 5 minutes. During those 5 minutes, you can go to your gel and wash the wells, which is really important because whenever you're making these gels, some of the polyacrylamide solution can still hang out in the well, and they have a different viscosity from your actual samples. And whenever you try to put a sample into a well that's not clean, your sample just kind of flies out of your gel. As your samples warm up, go back to your gel, stop the current, open it up, and just wash all the wells. So while your samples are warming up, we have to clean the wells. First, turn off the power generator by pressing the off button. Next, turn it off completely and get the anode and the cathode out of the box. So now we're going to open the gel box apparatus, slide it over to the left and pop it up. To wash the wells, I use the TBE that's already inside the first compartment of the gel box. And then I see the wells and then blast a little bit in there to effectively wash the wells. Do this about 2 times just to make sure that all the wells are nice and clean. This is also a really good time for you to assess which wells you want your samples to go in. Because of course, or when you take out the comb from your gel, sometimes you may not see the tearing of wells until you begin this part. And so it's important to assess if you have nice wells in order to accommodate your samples. In today's experiment, since I have 32 samples and 36 wells, I can only use a few of them and get rid of a few if they don't look good. The other thing to assess is to also look for bubbles that may have formed in your wells that weren't there originally before. So while looking on your gel over here, the potential sites you want to put your samples, see if downstream of that well there's an imperfection. So for example, I do have a couple of bubbles around some of these wells, but some of them are so far down that my gel will actually never run there, so I don't really care about that. This, however, over here is all the way to the side, so it's actually not that big of a problem. But when I see this bubble right here, an imperfection that I didn't see before, it actually is right in the middle of this well right here. So I will skip over that well because since the comb did have enough room for 36 wells, I don't have to use all 36 because I only have 32 samples. That's one way you can get away with it. Of course, it may not look as nice if you have to skip a sample in between, but you can always fix your image after you run your phosphor imaging on some sort of image fixing software. One of the other things that you'll want to do right before you put your samples onto the gel is that sometimes whenever you put on your samples, they can evaporate a little bit. And to combat that, once you start taking your samples off of the heat block, centrifuge them down for just a few seconds to get all that water that's evaporated up to the top of the cap downwards. Before adding a sample onto the gel, as you can see here, I've actually marked where I should not put a sample. And so I marked them by using this red Sharpie here, for example, and this will just make life more simple for you. You won't have to remember exactly what well didn't form very right or where the bubble is. As long as you mark it, you should be fine. What we're going to do is that we're going to use 6 microliters for every well. These wells can hold up to 8 to 10 microliters, but 6 microliters in each well for each sample should be more than enough for this assay. So now, before I add the samples onto the gel, let me talk a little bit about how I ordered these samples. So as you can see, I ordered the samples in columns, and in these columns you have the respective time points in order and which time points belong with which group by the superscript as compared to the subscript. Now I'm going to take a 6 microliter aliquot This can be thrown away into the radioactive waste. Going to find my first well, and yep. And again, because this is radioactive material, make sure you throw away your pipette tips into a container that's for radioactive waste, and the process repeats itself. So now we're going to restart the instrument. First, we have to close the instrument, put the anode and the cathode back to where they belong, turn on the power generator, and press the on button. So now it's reset itself. We'll see what voltage it will be running at. It's running a little over 1800, and this is already set to run for 5 hours. But of course, your gel will be a lot shorter, 2.5 to 3 hours. Since we preheated the gel and we heated the samples, it should go smoothly. Now that 2.5 hours have passed, you can see that the samples that were up here once previously loaded have traveled down the gel. The great thing is that for that one lane where we put an X, you can see that we have missed this bubble that appeared on the gel. Our samples have not penetrated through that. Another thing to know about the quenching solution that we used previously, it actually has two sets of dyes, this much lighter blue and this much darker blue-green color. This system was devised in such a way, this lighter blue band, as you can see, runs further on the gel than does this blue-green dye. This indicates that all your radioactive material should actually be in between these two dye fronts. What we'll want to do now is to deconstruct this whole gel apparatus. We'll have to drain out the TBE from the top compartment into a flask. But before that, we also have to turn off the generator in order to not electrocute yourself. Then after that, we'll get the gel out and pop open the glass plates in order to then image the gel. First, we're going to cut off the power. You can do this by pressing off on the generator itself, and you can see that all the volts, milliamps, have gone to 0. So then we also flip the switch in order for it to be completely off. Take off the anode and cathodes out, so there is no more current running on this gel. Now we can do the other steps. First, we're going to drain out that top compartment of this gel box. It comes with a tubing which you can insert into a backing that's found here in order for it to drain out into this bottle. All you have to do is put the tubing in and you will hear a snap. Once you hear that snapping sound, you should see the TBE from the top compartment flowing into the container down below. I actually noticed something was happening with the drain in the back. It wasn't draining properly, and so I decided to open the top of the gel box prior to complete emptying of the compartment. Sometimes whenever you take off the comb, they have little pieces of gel that are hanging around. If you notice that your system is not draining effectively, you might want to just open it up and see if there's anything clogging the drain, which in this case, there's still a few pieces of gel left up in there from when I took the comb off, and that should fix your issue. So now that the top compartment has been completely drained of the running buffer, we'll now have to get the gel out. I do this by slowly releasing the gel from the knobs. If you see any of the running buffer come through, that's completely fine, because now that your run is done, it's not as critical to not have any of this buffer on the back. You can just clean it off later by using water. Now we're going to take the gel, lift it out, and move on over. Now we're going to move over here to where I originally had these two round bottle flask holders. Now we're going to pop the gel open. So the best way to do this is to basically scraper and try to fit it in between the glass plates. This can get somewhat tricky. It's a lot better to do this quick whenever the gel's hot in order to get better separation. But as you do it, and if you siliconize your top plate correctly, you should see good separation of the top plate from the bottom. Should be able to either pick it up, pop the gel off. As you can see, perfect. No gel is sticking to the top plate. There's no gel on the siliconized glass. And all the gel is on the other piece of the glass. So in other words, we had a perfect separation and lift from opening the gel. So now the next thing to do is to actually get some parafilm a little bit over the length of the gel, and we're going to lay it over. So cut some parafilm. You can do this with scissors or a razor blade. I recommend scissors, they're just safer. If the parafilm wraps over, it'll be fine. You'll want to get the part that is easily outstretched. And now we're gonna lay that first portion over the radioactive part of the gel. You should be able to unfold some of this parafoam, even though it's crinkled up. And if you don't feel confident that you have parafoamed the important part of the gel, which is right in between these two die fronts, you can always, instead of lifting up the parafoam and redoing it, which I do not recommend, sometimes the gel will stick to the parafoam, and if you re-lift it up, it could tear your gel. And that would be a bad day. What you can do instead is get another sheet of parafoam and put it on the places where the gel isn't covered up. However, in this case, I got enough parafoam in both length and width that it covers the entire gel. And so now, to actually apply the film to perform phosphor imaging for the next day. Also, the other important thing to know is that the wrinkles on the parafoam don't matter. They won't affect the actual image that comes out. So now we're going to apply the phosphor imaging film. This film is actually found in this cassette that you can find by the Typhoon instrument, which is shared between us and the Harkai lab and the rest of the second floor. This is the film. The actual part that captures the radioactive phosphorus is this white side, but that's the side that we'll actually put down. As you may notice, this side on the right is our first sample that was labeled 1. Whenever you go to image this gel, you'll see that your results will look backwards, but don't worry, we have some software that will reflip the image in order for you to see your results the correct way. We apply this film right on top. You try to pat it down as nicely as you can, and then put some paper towels just to keep it a little darker, and to then not allow it for the film to move, we like to put some weight on it. We use the cassette itself. You can use other things such as these microcentrifuge holders to put some weight on it, but then basically you're done. This film has to stay on top of this gel for overnight, and then whenever you come back to lab the next day, just do the opposite, take off all the weight, and then put the film right back into its cassette. One important thing to note, you can do this even with light around, because of course the cassette will protect it from any sort of light that hits it. Even when you take it to the Typhoon, whenever you actually go and insert this cassette, you can see that it's magnetic, so the film is actually not attached to the bottom, it's attached to the top plate. As long as you move around with your film attached to this magnetic part, you shouldn't ever have an issue of worrying that the light will destroy your image. Now we get to deconstruct and throw away the gel. Since the gel is radioactive material, the gel must go into a solid waste bin. The best way that I do this, I use the actual saran wrap in order to pick up the gel. An easier way to do this, if you manage to do it this way, is to siliconize both glass plates. This will make your life a little bit easier. What I try to do is I use this scraper, which I use to pop the gel plates open. And I try to get as much gel as I can onto that saran wrap, roll up the gel in the saran wrap so it can come off nicely. Also, what you should know is that if you effectively take off this gel, there shouldn't be any radioactive material left on these glass plates. You can use a Geiger counter, which you should have on hand, to tell you that you have no more radioactive material on this gel plate. If there's ever a loose piece of gel, just keep scratching it off, putting it on top of the saran wrap. Then we throw away into the radioactive waste. I recommend you wash the plates as soon as that gel is off. Whatever little gel is left here on these spacers, it will harden over time. That makes it tricky to get off. So I would recommend that you wash this as soon as the gel is completely off. So here's the Geiger counter that I was talking about. Before you ever handle it, make sure that you do a swishing movement of your hands above the Geiger counter reader to make sure that you don't have any radioactive material. From those little chirps you heard, I don't have any radioactive material. When you should worry is when you hear a sound like that. That means that you have radioactive material that has fallen down either on your hands or on your clothes. Of course, if you point the detector right straight up into the ceiling, sometimes the instrument will freak out. As you scan the top of the gel, just a few inches off the top, you can hear that there's a tiny chirp, but there's none of this. So in other words, you have successfully taken off any sort of radioactive material that can be found on this glass plate. The next thing that you should do is actually test the running buffer that's at the bottom of the gel apparatus. Whenever you run your gel, the radioactive DNA and sometimes the radioactive phosphorus that you couldn't get out can run and will run faster than the rest of the material on the gel. And so it could have leached into this bottom TBE container. And if so, then that means we have to get rid of it in liquid waste that's radioactive. But as you can hear from the Geiger counter, there's no rapid chirping, so there is no radioactive material found in this TBE. So we can dispose of it just like any other TBE. We just throw it into HPLC waste, for example. Once we have the buffer out of the bottom component of this gel box, we rinse it out. Sometimes even if you leave a tiny little layer of what used to be there, it does tend to crash out and then you have these big salt crystals. If that happens to you, before your next run, you can just wash it with a little bit of water. It should readily dissolve, and then you can put it in a container and collect it, and then have this cleaned up, ready for next use. You should also clean the top compartment of this gel box. As you saw, there was some gel pieces floating around, so when I was trying to drain it, it did clog it up. So to prevent that in the future, clean up the top portion too. If there's any sort of salts here on the aluminum backing, just make sure that you clean it up too.